So it's my great pleasure today to introduce today's speaker, Dr. Sharon Murphy. Actually, Sharon and I were just talking. I think it's been almost 30 years that we have been colleagues and friends. She got her Bachelor's of Science degree in Chemistry and Biological Sciences at the University of Maryland, Baltimore County. She then got her PhD in Chemistry at the University of Colorado. She then went to American Health Foundation for a postdoc, and then went to University of California, Irvine for a second postdoc. And after that, she decided to come back to the American Health Foundation, where she started her professional career in the principal investigator track at the American Health Foundation, which was a wonderful nonprofit organization whose goal was to study the role of tobacco, among other things, in human health, and that's where she, Steve Hecton, Sharon, and I all got to know each other. So since then, she has become one of the leading experts in nicotine metabolism, which she's going to be talking about today. She's published over 150 papers. She's well funded. She has served as Director of Graduate Studies in the Department of, it's very long, Biochemistry, Molecular Biology, and Biophysics. I forgot to say that after the American Health Foundation, she moved here in 1996, where she is now full professor in that department. So without further ado, welcome, Sharon, and we look forward to your talk.
Thank you, Lisa. Yes, we've known each other 30 years, 1988. But we're not really that old. We were about 12 at the time. Okay. So I'm going to talk to you today about nicotine metabolism, smoking, and lung cancer, and specifically ethnic racial differences and the role of an enzyme, P450. I will often say P450-2A6 because that's the name of the enzyme, but the gene is CYP2A6, and everyone has come to call the enzyme CYP2A6 as well. So I'm sure I will use both terms. Okay. So before I talk about my work today, I want to just introduce a few things that I'm going to mention. Obviously, nicotine. I'll talk about nicotine metabolism, a biomarker that's referred to as total nicotine equivalence. I will define that for you and argue that it's a very useful biomarker in lung cancer studies. Briefly, I'll mention the important carcinogen NNK, and as I said, the enzyme CYP2A6 is going to be key to this talk, and that's the primary catalyst of nicotine metabolism. And then I'm going to look at the role of nicotine metabolism in smoking and lung cancer in two cohorts. One of these is the Shanghai cohort, and that'll just be some brief information there. And the majority of what I'll talk about is work that's been carried out in the multi-ethnic cohort study with subjects from that study. Okay, so nicotine is a major addictive agent in tobacco. Nicotine is a very simple molecule, but it's a very powerful molecule. Nicotine's not a carcinogen, but nicotine is why people smoke. Take out the nicotine, and they really don't like cigarettes much. Okay. The hypothesis that's driving the work that I'm going to present to you today is that lower levels of nicotine metabolism will result in decreased smoking intensity, and therefore will decrease a smoker's exposure to that tobacco as they will smoke that cigarette less intensely or smoke fewer cigarettes, leading to a decrease in lung cancer. The carcinogen that I'm going to use as a carcinogen, a measure of exposure to carcinogens, over 7,000 compounds in tobacco. Among those, more than 70 have been identified as carcinogens, but NNK is the one I'm going to focus on. We think that's an important one. One of its strengths is it's specific to tobacco products, so you can't be exposed to NNK unless you use a tobacco product. It is carcinogenic in humans, as defined by IARC, the International Agency for Research on Cancer. It's considered one of the major causes of lung cancer in smokers, and of importance to this talk, there's a metabolite, NNAL, which is a well-established biomarker of NNK exposure. So levels of NNAL in the urine of a smoker will reflect the amount of NNK to which that smoker was exposed. So first, before I talk about our work, I just want to give you a little update or background on why we really care about smoking still. Cigarette smoking and lung cancer mortality in the United States is still an important public health problem. There are over 40 million smokers still in this country. 90% of lung cancer is caused by smoking. Lung cancer is the leading cause of death in men and women. And in 2017, was predicted, I don't know if it matched up to the numbers, but it was predicted that 155,870 lung cancer deaths would occur in the United States. However, lung cancer prevalence is not equally distributed across the United States. So if we look around this room, probably no one smokes, so we never run into smokers. And even in Minnesota, the prevalence is relatively low, being 12 to 14% of Minnesotans smoke. However, if you move to Kentucky or Tennessee, that number is more like 20 to 25%. And if you look at a map similar to this with lung cancer deaths, it's going to mirror it exactly. Okay? So cancer smoking prevalence has been decreasing for the last 50 years, ever since the Surgeon General's report came out in 1964. I think that's right. Okay, so 54 years. However, it's still the major cause of cancer death in this country. Excuse me. One of these buttons is, okay, one of these, I don't have a pointer. I did have it. Oh, there we go. Okay. Where the red line is lung cancer deaths in the U.S. from 1930 through 2014 for men and women. So as you can see, it's still a major cause of cancer deaths. Worldwide, the problem's even bigger. There are 820 million men who are daily smokers in the world. In the last 30 years, there's been a modest decrease of about 10% in that number. Only about 176 million women are daily smokers, so women are a little smarter than men. But female smoking is more common in high-income countries, so I don't know what that says to us. So it's a problem for both men and women across the world. That's just illustrated here to give you a flavor of the distribution of smoking prevalence in the world. You look at something like the U.S., no longer. If you look at Russia, the prevalence is very high, whereas in the U.S., on average, it's quite a bit lower. The prevalence, of women is somewhat different. As I said, it's concentrated in the higher socioeconomic countries. So worldwide, there are over a billion smokers. 80% of lung cancer in males, 50% in females is caused by smoking. Leading cause of death in men, second leading cause in women. 1,589,000 lung cancer deaths in 2012, or 3 every minute. So a lot of people die from lung cancer. 11% of female, 24% of male lifetime smokers will get lung cancer by the time they're 85 years old. So it's a problem still. So what is the relative risk of lung cancer? We kind of lose track of this because we know smoking causes cancer, but it's a big problem because the more you smoke and the longer you smoke, the greater your risk. So if you've been a lifetime smoker, smoking for 50 years, a pack a day, your risk of lung cancer is 20-fold, your relative risk. So how does that compare to something else that causes cancer in non-smokers? Radon is known to cause cancer in non-smokers, and that relative risk is 1.3. So the relative risk from smoking is orders of magnitude greater than most compounds that we're familiar with that cause in a human population causing cancer. However, that risk varies by ethnicity. And this is the beginning of a large study that we began a few years ago. This is a report in the New England Journal of Medicine in 2006 that compared the lung cancer susceptibility African Americans, Native Hawaiians, Latinos, Japanese Americans, and whites, and looked across different reported cigarettes per day. And if we just look at where this difference is greatest in exposures of reported less than 10 cigarettes per day, if this is normal, oops, backwards, forwards, okay. I shouldn't put this on the same. This is normalized to African Americans with a relative risk of 1. Whites have about half that risk. Japanese Americans a quarter the risk, and Latinos even less. So this publication came out in 2006 from investigators. I can't hold two things at once. This report came out in 2006 in the New England Journal of Medicine. And it was, I'm sorry, I had this. You can look at this another way, and I'm going to come back to this slide later. You can look at this another way if you look at the rate of lung cancer across these ethnic groups. Here in the middle are the white smokers, European Americans, they're described here. The highest rate is for Native Hawaiians and African Americans, and the lowest rate is for Latinos and Japanese Americans. So this work was carried out in the multi-ethnic cohort, which is a prospective epidemiology cohort of genetic lifestyles relationship to chronic disease, over 200,000 men and women, recruited at the age of 45 to 75 in 93 to 96. And they were recruited from the Los Angeles area and from Hawaii. The major organizations involved here are USC in Los Angeles and the University of Hawaii in Honolulu. The 5 groups which were followed, and of importance to our study, a subset of these participants, 2,393, which is referred to the subcohort, which have biospecimens, have urine and blood samples, and that was of interest to us because then we could look at biomarkers in these individuals. So what that led to is a collaboration between the investigators in Hawaii, Louis Lemachon and Lonnie Park, and the University of Southern California with Chris Heyman and Dan Stram. And that began this program, large program project grant. The PI is Steve Hecht, where we're looking at the mechanisms of ethnic racial differences in lung cancer due to cigarette smoking. Now, the hypothesis that was driving that PPG was that ethnic differences in the exposure and response to tobacco smoke carcinogens contribute to the observed differences in lung cancer risk. So the differences that were reported in 2006, African Americans highest rate, Japanese Americans lowest rate, it was our hypothesis that some of that is driven by differences in exposure and how each smoker handles the carcinogens that are present in that tobacco. So the accepted mechanism of how tobacco carcinogenesis works, beginning with smoking addiction, you smoke, you get a lot of carcinogens, they can be activated, they can bind to DNA, you can activate important genes, ultimately get lung cancers, there's repair pathways, there's detoxification, there's lots of things that can be influenced in this pathway. So any one of these could be varied across these ethnic groups. All of them are influenced by components of tobacco. However, I don't care about any of that. All I care about is nicotine and step 1. Because if there's no nicotine in a cigarette, they're not going to smoke it. So if we don't understand how people handle nicotine, all the rest of it doesn't make any sense. It isn't important. So nicotine, major addictive agent in tobacco. So now that leads me to a very simple-minded scheme, which is much easier for me to handle. So you have tobacco, you consume nicotine, you get addicted, you keep smoking, you keep smoking, you keep smoking, and ultimately you have a risk of developing lung cancer. This can be interrupted by the metabolism of nicotine. Nicotine gets pulled out by conversion primarily to a metabolite referred to as cotinine. So the hypothesis that drives a couple of projects in this program project grant and also is driving the work that I'm going to present to you today is that differences in nicotine metabolism across ethnic racial groups result in a variation in smoking intensity and therefore carcinogen exposure per reported cigarettes per day. And that these metabolic differences will contribute to the ethnic racial differences in lung cancer. And hopefully after I present the data today, you'll believe this hypothesis is true. So this raised 3 questions that I wanted to answer. One is there a biomarker that accurately reflects an individual's tobacco smoke exposure? Something better than CPDs, cigarettes per day. Does variation in nicotine metabolism influence nicotine intake and tobacco carcinogen exposure? And the big question, does variation in nicotine metabolism influence a smoker's risk of lung cancer? So question 1, is there a biomarker that accurately reflects exposure, and does it influence these observed differences in lung cancer risk? And I wouldn't ask the question if I didn't know the answer. The answer being yes. That marker that we believe is better than cigarettes per day is total nicotine equivalence. What is total nicotine equivalence? That's nicotine plus 6 of its major metabolites that are excreted in urine. And by measuring those 7 compounds, I can account for 85% of the nicotine dose, and therefore, that's an excellent marker of tobacco smoke exposure. So how is nicotine metabolized? So nicotine is metabolized by 3 pathways. Nicotine N-oxidation through an enzyme FMO3, nicotine glucuronidation through UGT2B10, and nicotine C-oxidation, which goes through 2 steps, P450 or CYP2A6 catalyzed, generating cotinine. Each of these is converted to glucuronides, and cotinine is also converted to 3-hydroxycotinine. We can accurately quantify all 7 of these metabolites in a smoker's urine, and that's all these in these boxes here. And so if I measure nicotine N-oxide, nicotine glucuronide, cotinine glucuronide, trans-3-hydroxycotinine, and trans-3-hydroxycotinine glucuronide, and I add those all up, I can account for more than 85% of the nicotine dose to which that individual was exposed, and so I can get a measure of exposure in each person. So we did that in the multi-ethnic cohort for the 2,300 smokers for whom we had urine samples. And in this graph, I'm looking at the geometric means of total nicotine equivalence for each of the ethnic groups. And these ethnic groups are listed in the graph in the order of their relative cancer risk. So the highest cancer risk is for African Americans per cigarettes reported, and the lowest is for Japanese Americans, and intermediate, the other 2 groups. So there's a statistically significant difference between total nicotine equivalence in African Americans compared to whites, and between whites and Japanese Americans, and that's in the direction of the cancer risk, meaning African Americans are exposed to a greater amount of total nicotine equivalence than are whites, and Japanese Americans exposed to a much lower concentration of nicotine equivalence. Stepping back a minute, so the data that I just presented here is this data. African Americans, 44 nanomoles per ml nicotine equivalence, whites 36 nanomoles per ml, and Japanese Americans 27. However, their reported cigarettes per day is completely different than that. African Americans reported cigarettes per day was 10. Japanese Americans were 12. So clearly the exposure from 10, the average 10 cigarettes in African Americans is much greater than the 12 cigarettes reported for Japanese Americans. So in addition to total nicotine equivalence in this population, total N-al was measured, remembering that N-al is the metabolite of the tobacco-specific carcinogen NNK. And N-al levels mirror the total nicotine equivalence, meaning that the level of N-al in African Americans is twice that in Japanese Americans, even though the reported number of cigarettes is quite similar, and whites are intermediate. And that's illustrated here. Again, African Americans are statistically higher exposure to NNK as measured by the biomarker N-al, and that in turn is higher in whites than in Japanese Americans, consistent with the risk in these 3 ethnic groups. So yes, we have a marker that seems to reflect better the exposure of the groups. Now the question is, does variation in nicotine metabolism influence that intake that we've just measured in carcinogen exposure in this groups? And that becomes a little bit more complicated question. So again, there are 3 pathways of nicotine metabolism. So 3 enzymes, FMO3, UGT2B10, and P450 or CYP2A6. But by far the major pathway in the majority of smokers is P452A6 catalyzed 5' oxidation of nicotine, ultimately generating cotinine and 3-hydroxycotinine and their glucuronides. If I quantify, which we have already, nicotine N-oxide and all these metabolites, I can measure all the products of 2A6 metabolites by summing these compounds, and I can measure the glucuronide and the N-oxide for each of the 5 ethnic groups. So if I measure all 3 pathways for each of the 5 ethnic groups, I've displayed them here in the proportion of each of these pathways for each of these groups. So the blue, the dark and pale blue, are products of C oxidation, so that's cotinine and 3' hydroxycotinine. Green is nicotine glucuronide. Yellow is nicotine N-oxide. And the red is the nicotine that's unmetabolized, left in the urine. And there are 3 things I want to draw your attention to here is the amount of nicotine, percentage of nicotine left non-metabolized in Japanese Americans versus, for example, African Americans or whites is at least double that. In addition, the green bar, which is glucuronidation, is much lower in African Americans than it is in the other groups. I'm not going to talk about that today. That's a seminar in and of itself. But the third thing is that overall, C oxidation in Japanese Americans and Native Hawaiians is much lower than it is in the other 3 groups. And Japanese Americans excrete the most unmetabolized nicotine. So the question becomes, does lower CYP2A6 catalyzed C oxidation in the Japanese Americans lead to the lower total nicotine equivalence that I presented to you at the beginning? And then what is that relationship in those other ethnic groups? So remembering, P452A6 or CYP2A6 catalyzes that reaction, converting nicotine to cotinine. CYP2A6 activity varies significantly across people. So one person may have 2 or 3 times greater activity than another person. And that's predominantly due to the large number of variants in this enzyme that exist. More than 50 variant alleles have been identified, not characterized, but identified. One of which is a deletion, which is referred to as CYP2A6*4, and this is quite common in Asian populations. Okay? So there is a subset of Asian individuals who have no 2A6 activity, so really don't metabolize nicotine much at all. So I have to tell you one more thing. So that's 2A6 metabolism driven by nicotine metabolism driven by 2A6. 2A6 also catalyzes the conversion of cotinine to its metabolite, 3-hydroxycotinine. So we can use the ratio of 3-hydroxycotinine to cotinine because those compounds are more abundant and longer lived than is nicotine as a phenotype of P452A6 activity. So if in a smoker I measure 3-hydroxycotinine levels in the urine and cotinine levels in the urine, and I establish the ratio of the 2, that will be a reflection of CYP2A6 activity. And again, we did this in the 5 racial ethnic groups and demonstrated that Japanese Americans have a much lower CYP2A6 activity than do African Americans, whites, or Latinos, and Native Hawaiians are somewhat intermediate in that. And in part, that wasn't really a surprise because we knew there was a high prevalence of the deletion in Asian population, Japanese, and to some extent Native Hawaiians, although that had never been looked at before. Now we have to figure out what genes are driving this. So what CYP2A6 genotypes contribute to these differences across the 5 ethnic groups? So looking in the literature and some of our own work, we chose 10 SNPs and the *4, which is a copy number variant, the deletion, based on what we already knew about their CYP2A6 activity. And we genotyped these across these 5 different groups. And now these pies are reflecting the frequency of these various haplotypes, which are no alleles. Green is good, lots of metabolism. Red is no metabolism. This is the deletion. And then all the intermediate alleles in between have intermediate levels of metabolism, either due to differences in the enzyme active site, or in the case of, for example, star 9 is a Tata-box variant, so there's less expression of the enzyme, so you have less protein. Okay. Using this, we characterize genotype in all these 5 groups. Okay, but we have 2 alleles of every gene, so there are diplotypes. Okay, there are 68 possible diplotypes in those 2239 smokers. So in order to categorize these individuals, we define 6 diplotype categories by, again, putting each of the haplotypes into 3 categories: normal, meaning no variant allele or no effect on activity; intermediate, some effect on metabolism, or little or no activity. And using each of these, then each person has to be either homozygous or heterozygous for one of those alleles. So they're either NN, completely normal, NL, NI, or LL, meaning they have loss of allele, loss of activity, null with regard to P450-2A6 activity. We defined everybody by these diplotypes. First, we just looked at the ethnic groups without the ethnic groups, just making sure our definitions are reflecting CYP2A6 activity relatively accurately in this population. So what I'm looking at here is the geometric mean of CYP2A6 activity. That's that ratio of 3-hydroxycodenine to codenine in these 6 different groups, normal to null. Okay? And as hoped and predicted, the activity decreases as you go from normal 2A6 to low-level 2A6 activity genes. Gets a little more complicated when you start to look by ethnic group. Okay, this is a very busy slide. The colors are the different groups. The African Americans, again, they're in the order of their cancer risk. African Americans having the highest reported lung cancer risk, down to Japanese Americans, the lowest risk. Okay, and each of them go down with these different diplotype categories. However, better or worse in some of the groups based on, in part, the numbers. So the frequency of each of these variants varies across these ethnic groups. And also, we haven't defined all the variants that exist in P450-2A6, so we're missing things. However, for African Americans and what we've measured, there's a reasonably good decrease in activity as we go from normal to intermediate and loss of activity alleles. Whites, the relationship isn't quite as good. There are only 6 people in that LL group, and clearly in some of the intermediate groups, we're probably missing some genotypes that would better define these activities. But in the Japanese American, we have a nice step down from normal activity to loss of activity alleles. And now what we've done is characterize the CYP2A6 activity in each of the ethnic groups. But what's the question that we're trying to ask? The question we're asking is, does this activity influence exposure to tobacco smoke? So are total nicotine equivalents influenced by this activity, and is carcinogen exposure influenced by this activity? So we looked at total nicotine equivalents by these diplotype groups. Okay, and on the right of this, which is highlighted or illustrated here, this is Japanese Americans, where the green, no variant alleles, red, all loss of homozygous for loss of function alleles, total nicotine equivalence decreases exactly the way the CYP2A6 activity decreased. It's important to point out that there are only, out of about 700 Japanese Americans, only 31 had no variant allele. So almost all Japanese have some variant 2A6 allele, and that's influencing not only their P450-2A6 activity, it's influencing their total nicotine equivalence. If we look at 2 other groups, the African Americans and the alleles that we're looking at, there is a decrease across These groups, again, for total nicotine equivalence, mirroring the activity of CYP2A6 activity. In whites, that relationship isn't that good, but it mirrors what's going on with CYP2A6 activity. I want to point out here, if you look at those 31 individuals here, Japanese Americans, total nicotine equivalence is the same for those 31 individuals, which is only about 5% of the population, as it is for the other groups. So that would suggest within that, if we pull out only the Japanese Americans that have no variant alleles, their cancer risk will probably be similar to those of African Americans or whites with regard to carcinogen exposure. We did the same thing looking at N and Al. Remembering N and Al is the metabolite of the tobacco-specific carcinogen NNK. This is looking at N and Al concentrations in the urine of these three ethnic groups by diplotype. Again, normal, all the way to loss of allele. Parallels exactly what's going on with CYP2A6 activity, total nicotine equivalence, and N and Al being NNK exposure. So does variation in nicotine metabolism influence nicotine intake and tobacco carcinogen exposure? Yes. Yes. I think that we've showed this in the African Americans and Japanese American groups, which is what this says here. Japanese Americans, African Americans, CYP2A6 diplotype, decreased total nicotine equivalence, decreased N and Al. Smokers in these two groups who have low activity alleles have lower levels of total nicotine equivalence and N and Al. We didn't find a significant association of T&E or N and Al with diplotype in these other groups, but in part that's due to the low frequency of the genotypes that we did determine. And in addition, I wrote there may be uncharacterized variants. There are uncharacterized CYP2A6 variants that are going to influence metabolism that we haven't been able to quantify. This is a pretty complicated gene, actually. And there also may be other environmental or genetic factors that contribute significantly to this activity and nicotine uptake in these other groups. But where there's a lot of variation in 2A6, that's a major player in the tobacco carcinogen exposure of smokers. So if we put this all together with regard to cancer risk in this multi-ethnic cohort, African Americans, more nicotine taken in per cigarette, more carcinogen per cigarette than whites. That's consistent with their higher lung cancer risk. That difference is not completely driven by nicotine metabolism, so there have to be other factors contributing to those observed changes in exposure. Japanese Americans take in less nicotine per cigarette. Than whites, consistent with their lower cancer risk. And this is driven a lot, if not predominantly, by lower nicotine metabolizing 2A6 forms prevalent in that population. So now that leads us to question number 3. Does variation in nicotine metabolism influence a smoker's risk of lung cancer? So what do I know about CYP2A6, the gene in lung cancer? The argument that CYP2A6 is associated with lower susceptibility to lung cancer has been around for more than 20 years, probably. The CYP2A6 star 4 deletion was identified about 20 years ago, and it has been shown in a number of relatively small studies. Lower susceptibility to lung cancer does occur in Asian populations. However, data to establish a direct link between nicotine metabolism, smoking dose, and lung cancer is really very limited. Recently, in collaboration with Jingming Wan, now at the University of Pittsburgh, we determined the influence of CYP2A6 genotype on nicotine metabolism, total nicotine equivalence, and lung cancer in the Shanghai cohort. So what's the Shanghai cohort? 18,000 men aged 45 to 64, recruited in 1986 at baseline at that point in time, prior to cancer incidence. Blood and urine samples were collected. Incident lung cancer cases have been identified in the subsequent years. And we evaluated CYP2A6 activity. That's the ratio of 3-hydroxycotinine to cotinine as a measure of nicotine metabolism in 400 and genotyped for some of the common alleles in Asians in 475 lung cancer cases and 475 controls. What's illustrated here is the relationship between the CYP2A6 genotype or diplotypes. On the side, we only did 4 of the most common variants in the Chinese population, and we're looking at the relationship of genotype to phenotype. So we're looking at the relationship of these alleles to total 3-hydroxycotinine-cotinine ratio, and by doing that, we identified groups which we referred to as poor metabolizers, again, in this case, slow and intermediate metabolizers, and normal metabolizers. Then to look at cancer risk, we use these 4 categories. So in this cohort, if you look at the relationship with the risk of lung cancer in poor metabolizers, individuals who carry deletion alleles, versus the other 3 groups, the relative risk, or the odds ratio in this example, is 0.64 for that group versus normalized to 1 in the other groups. And that's statistically significant. However, if you adjust that, okay, so here is the relationship unadjusted for total nicotine equivalence. If we adjust these values for exposure based on total nicotine equivalence, now there's no longer a statistically significant difference between the different metabolic groups. Okay? So CYP2A6 poor metabolizer is related to low cancer risk, but this association disappears after adjusting for total nicotine equivalence. And as far as we're concerned, that's a good thing, because what that does is establish the link between CYP2A6 genotype and total nicotine equivalence, which supports our hypothesis that variation in nicotine metabolism influences lung cancer risk due to its effect on smoking intensity. Okay. So what about in the multi-ethnic cohort? Is CYP2A6 activity or genotype associated with lung cancer? The answer is yes. But we did it a little bit differently. Okay. So in the multi-ethnic cohort, a genome-wide association study was carried out where the outcome was biomarkers. Okay, so genotypes were identified that were associated with CYP2A6 activity in the MIC. Okay, in the sub-cohort, the 2,300 whom we had urine from and could do biomarker analysis in. Those same, and I'm going to tell you this whole story. This is this conclusion first. Those same SNPs that are present in the GWAS of metabolism in the MIC study were associated with lung cancer in a large GWAS consortium, which we carried out collaboratively. That consortium is referred to as TRICKLE, Transdisciplinary Research in Cancer of Lung Consortium. And the database consists of 13,000 cases and 43 controls, lung cancer cases for whom there was GWAS, genome-wide association data. These individuals are all of European descent. Okay? So now we don't have the benefit of the high prevalence of the deletion allele. That's true in the Asian population. So how was this done? So in the multi-ethnic cohort, a genome-wide association study was carried out with the biomarker data. So looking at single nucleotide polymorphisms across the genome and how they relate to particular biomarkers. There are 11 million SNPs included in this study, either from genotyping or imputation. And we observed that both CYP2A6 activity, that's the relationship of 3-hydroxycotinine to cotinine, and total nicotine equivalence were associated with a signal on chromosome 19. Chromosome 19 is where P452A6 or CYP2A6 is. And that's illustrated here in what's referred to as a Manhattan plot. And what you're looking at here is the chromosome number and the p-value for the association between a single nucleotide chain on that genome versus on a particular chromosome. And as you can see, all of those relationships are in chromosome 19 with p-values up to 10 to the minus 48. So these are the SNPs that I'm talking about that have been identified in the mixed study looking at metabolism versus GWAS in a GWAS. And then the question, are those SNPs associated independently with lung cancer? So there were 248 SNPs. That was 13 independent signals. A lot of those are linked to each other. They travel together, okay? Associated with CYP2A6 activity in the MYC. So that was all our data. 226 of those SNPs were also available in the database of the Trickle study. Among those 226, 6 were globally and significantly associated with lung cancer. Okay? So SNPs that were identified as related to nicotine metabolism were subsequently identified as related to lung cancer risk in this large cohort. So these data support the hypothesis that greater CYP2A6 activity which results in increased smoking and greater exposure to carcinogens, does result in an increased risk of lung cancer. And that study was carried out collaboratively. I've only listed a few of the authors, but there are about 20, led by Chris Amos at Dartmouth on the consortium. The variants that we've identified here may now be used as risk prediction markers in smokers. But these SNPs are not causal variants, meaning they don't influence metabolism directly. They're markers, okay? And the exact causal variants are yet to be identified, which may be multiple variants that are being tagged, so to speak, by a single SNP that's not affecting directly metabolism. So in summary, CYP2A6 genotype is a biomarker of lung cancer risk due to its effect on nicotine uptake and tobacco smoke exposure. Low CYP2A6 activity results in lower cancer risk. We believe that total nicotine equivalence, CYP2A6 genotype or activity, combined with other genetic polymorphisms, for example, polymorphisms in the nicotine acetylcholine receptor, which is important in how people experience nicotine. Okay, when nicotine comes in, the receptor binds it, and that's going to influence your response to nicotine. So clearly that's going to have an impact on smoking as well. Combining that as well as other measures, we may be able to produce an algorithm for predicting lung cancer susceptibility in smokers, and that could ultimately be useful in lung cancer screening. So I'd like to acknowledge some of the people involved in this work. In my lab, all of the total nicotine equivalents were carried out by Elizabeth Thomas and Nicole Thomas, working with Linda von Weymarn. The NNL data is from Steve Heck's lab, with Steve Carmella put together all that data. At the University of Hawaii, there's Louis Lemarchand, who is a very talented epidemiologist. I think in some ways an unusual one because he really understands metabolism, which is very refreshing for me. Marit carried out all the genotyping on CYP2A6. The University of Southern California, on many of the references, you saw Park. That's Lonnie Park. I've worked closely with Lonnie Park at USC. She began as a postdoc with Louis and is now an assistant professor at USC. And is a wonderful epidemiologist and collaborator who understands what I say before I say it, which is always nice. And then Chris Heyman, who was on the original publication, is a geneticist at USC. Dan Stram and Yesha Patel are the statisticians that have been wonderful to work with. And the support is from the National Cancer Institute and particularly, specifically, the PO1 mechanism, ethnic racial differences in lung cancer due to cigarette smoking. I'd like to thank everyone for their attention. And I'll take any questions.
Do we have any questions or comments?
So, Sharon, can I make sure I understand the implications? So Japanese Americans don't need to smoke as much to maintain the free nicotine in their bloodstream.
Correct.
But if I understand Dorothy's work correctly about low nicotine cigarettes, you can get people to quit if you don't give them enough nicotine. So kind of two related questions. Why do African Americans bother to smoke if they can't maintain free nicotine levels? And secondly, does the CYP2A6 polymorphism make a difference in sort of the low nicotine quit trial, so to speak? I would imagine people who can extract enough nicotine out of a low nicotine cigarette, they won't be reinforced to quit because they're fine with the amount of nicotine contained in that cigarette.
Right. So the first question was African Americans. I already forgot.
Yeah, so again, I'm trying to...
why do they smoke?
Why
Okay,
do they smoke if they can't maintain a
They
free
can
nicotine level? Why don't they
enough.
just give up?
They can maintain enough.
Okay, so
Okay,
there's
we're not.
enough nicotine in
Yeah, if you look at circulating cotinine levels, which is a measure of actual constant exposure, because smokers tend to maintain roughly the same amount of cotinine, which is the metabolite of nicotine. Nicotine goes down quickly. It's similar in African Americans to other groups. Okay, so yes, they can maintain enough. The second question is a really interesting one, and one that actually came up at our recent meeting of the program project grant. Luik said something about the low nicotine cigarettes and how that's exactly like being Japanese. So the low nicotine cigarettes, though, the answer to the question is the nicotine is so low in those cigarettes, you can't get enough. I mean, I always say when, we measured a lot of these total nicotine equivalents in these people who are smoking these low nicotine cigarettes, and when the number was just a number, greater than, say, 5 nanomoles per mil, and I said, there's no way they're smoking these cigarettes. They'd have to eat them to get that much nicotine. So I don't think you can get enough nicotine from those cigarettes. And it would influence you asked about the metabolism, and obviously that would have an influence on smoking, the amount of those cigarettes.
Any other questions? I'll ask a question then. So you focused on the African Americans, the European Americans, and the Japanese Americans, but it seems like the Latinos and the Native Hawaiians don't really fit exactly the same, and I was wondering if you could speculate as to why they don't fit that need. The same story. What do you think is going on in those populations?
I have no idea. But the story in the African Americans and the Japanese Americans is different, too. Because in the Japanese Americans, the nicotine metabolism is driving the whole, when you look at the group as a whole, it's driving that mean lower level of total nicotine equivalence. When you look in Japanese Americans, when you look in African Americans, when you look at the groups who have lower nicotine metabolism, they do have lower total nicotine equivalence. But as a group, total nicotine equivalence is still high in African Americans, independent of the change in metabolism. So something else is, I don't use the right word, encouraging African Americans to smoke more. With regard to the other two groups, and we're always comparing to whites, like we're the middle, but the other two groups, the Native Hawaiians, with regard to metabolism, they're close to Japanese. So again, as in the African Americans, that's not having a big impact on their smoking, and I can't tell you what is. Okay, and Latinos, to be honest, I haven't thought about much at all, other than I think the smoking levels are a lot lower as a population in Latinos, but I think that's actually changing. So I don't have an answer. Yes.
So you mentioned that in the African Americans, you thought that behavior might be differences in behavior might be associated with why they don't smoke more cigarettes, even though they're not getting the same nicotine from each cigarette. So I was thinking perhaps this has something to do with dopamine or dopamine metabolism, dopamine uptake. Are there differences in African Americans?
There's some data. There's data on the receptors too. There are variants in the nicotine receptors that occur in African Americans that don't occur in other ethnic groups. The data on how that's linked to exposure and what's actually been looked at is addiction studies and looking at those. They're not that tight, so to speak. I mean, they don't hold it up well, but the possibility that that's contributing is certainly, there are variants, so you can make the argument that that could contribute. But I think it's a myriad of things, probably. So.
Any other questions? Okay, well, with that, let's give Dr. Murphy a round of applause for her excellent presentation.